Hello everyone, and welcome to today's webinar, the Cytobank Solution, a cytometry data sharing platform for collaborative multicentric data analysis. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more, visit beckman.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Dr. Hervé LUCHE, Director of the Immunophenotyping Core of CIPHE. Dr. LUCHE, you may now begin your presentation.
Many thanks for this kind introduction and good morning. Hello everyone. So I'm going to present you this webinar on the Cytobank solution and how we use it at the Center of ImmunoPHEnomics to drive our collaborative multicentric data analysis. So first, a disclaimer that this presentation is sponsored by Beckman Coulter and that the content is scientific, non-promotional, and educational, and that the views are actually mine, not controlled by Beckman Coulter, and that Beckman Coulter does not accept any responsibility or liability for the accuracy, content, completeness, legality, reliability of the information presented here. So first slide that I wanted to show is this one. It's to my view the core elements of the Cytobank platform and why it's of interest to all of us. First, it's a biologist-friendly interface, so you don't need any bioinformatics skills to actually use it and drive your analysis. Next, you can do data sharing and also analysis sharing, which is very interesting. We will see how we can do that. It includes tools for data pre-processing, which sometimes is a bit tricky to master on other platforms. And you have embedded machine learning algorithm. And this, most of the time, is actually what is being presented and put up front in webinars. I'm not going to do that today because I would like to show you the powerful of the interface in terms of organization of the experiment, as well as the illustration that you can build with this solution, so that you can build efficient visualization of multi-group data set, and also that the Cytobank platform allows for integration of data partition, meaning clustering, generated by algorithms that may be not present in Cytobank platform, but you can still use to actually visualize your data on it. And very importantly, it's a tool that allows you to keep an eye on your cell scale, which is important for investigation of the quality of the clustering and partition of your data, because sometimes you really need to look. You might have some difficulties there. And finally, you can extract statistical table with a large panel of metrics that could be useful for your project. So let's start first with a presentation of where I'm working, so that you understand better why we went to adopt this solution at CIPHE. So what we basically do is cytometry in all kinds, so flow, spectral, mass, as well as genomic cytometry. And the idea of what we do is to actually immunoprofile the immune system of mostly mouse models in different settings, in disease, immuno-oncology, it could be inflammation or infection, to better understand how the immune system is perturbed in presence of a challenge, and also our treatment candidate could actually improve clinical outcome. One example is MC38 tumor model. So we could do a clinical efficacy like a lot of labs do, but what we really put our efforts is actually extracting cells, immune cells from different organs, lymphoid organs, actually, as well as tumor mass, and use the different technology we have at CIPHE to actually immune profile in depth the immune system. And then experiment could look like this with different panels. And really the goal of this is to be able to do this analysis in unsupervised manner, try to be as descriptive as possible to identify populations. And then next is to actually correlate a change in density of population that correlates with treatment and obviously a treatment efficacy like what we see here. We see that the dose, at a very high dose of this XXY treatment, we see a complete change of the immune profile that we would see with isotype control. And to do this, actually, we work fully in a panel that is done at CIPHE, or we actually work with externalization of our panels that's possible in mass cytometry from this technology published in Cytometry A. The idea there is that we combine interactions, so we send our panels to our partner, they use them in their workflow and then send us back stained cells, and then we do the analysis for them. And then you start to understand why we need Cytobank for. The data analysis and raw data need to be analyzed by us, but shared by our distant partners. And we'll see how we do that. What we also use Cytobank for is to project our in-house analysis capabilities to partner sites. So we have in-house a suite of different tools for pre-processing, dimensionality reduction, and clustering annotation. And we use Cytobank to visualize, enrich FCS file, and I will come into in a minute how we do that. So the Cytobank platform usage at CIPHE is really to be able to project our analysis capabilities to distant partners, to also do remote validation of newly generated data sets. So if I have my collaborator generating data sets, I can have a look at the data through Cytobank and also give advice on the analysis perspective. We can use Cytobank to visualize results of data partitioning and check whether clustering went okay. We can visualize results very quickly of multi-group, multi-sample data set and interrogate on the fly data set during collaborative data reviews. We will see that. And also we can use metadata tags, which is very important to actually organize outlayers illustration on Cytobank. So now if I go in the different part of the platform I would like to showcase today, I will first talk about the experiment manager because it's a nice way to organize your data, to actually show to actually show that I will use a data set that we have generated throughout a consortium, which is CELFEDIA. CELFEDIA infrastructure is actually an alliance of 4 research centers that have complementary expertise in animal experimentation. So the CIPHE is there from is mouse expertise, but we have a center that is really focused on rat immunophenotyping, knowledge that we don't have. But we have, there is another site like us, ANIRA, who is trained for advanced data analysis methodology. So the idea is really to join forces to push immunophenotyping within this consortium. But for this, you need to have a plan, and especially a plan on how to organize your data, and that's what we did on Cytobank, using a dedicated license for the CELFEDIA work. And to sum up the big challenge we had, it was the data management, because within this project, the Parcel project, we had several working packages, and each time we needed to share data and analysis so that we can draw on conclusion in a collaborative manner. And that has been difficult because we don't have, at the consortium level, an IT infrastructure. Next, we also need to work along the FAIR principle, which are findability, accessibility, interoperability, and reuse of digital data sets. And for this, we needed to find a solution, and Cytobank for us was the best available. And then how to share raw data sets and corresponding analysis in a curated, searchable manner, and then Cytobank was very good for that. Why? Because we could basically define labels to actually tag all of our several experiments. So to recall, in this consortium, we deal with mouse and rat immunophenotyping. We have flow and mass cytometry data set. There are 4 work packages. There are some experiments that are designed for setup and other for actual analysis of samples that we need to deliver results for. So you need organization, and we did this through this sequential. So in Cytobank, you have the capacity to put labels, like you see here. You can create a new label, like here. I created one VISNI and tagged 2 experiments with the VISNI label so that I can use that later on for my investigation of the data sets. then next, what I can do is basically use quickly this filter. So if I go on, for example, here, BI, I can actually filter all the experiments that have this label and show them in the interface. So like this is very efficient to find an experiment that has been stored on Cytobank, and that's why we needed this common language for the consortium so that everyone can use these tags to go through the data set very efficiently. Next, once you have selected an experiment, you come to this window, which is an experimental detail of the experiment and what is there. So you see that we have an experiment summary with the different labels. We also have a part where we see illustrations that have been developed. FCS file included into this experiment. So we see here 156. It's a lot. So you need a plan to visualize data from that. And then we have attachment. And I wanted to draw your attention that in the attachment you can put illustration results or PDF and images, but you can also put all related documents to the experiments like SOP, papers describing the work that is being done in the experiment, or a sum up as a PPTX format to actually show the results. And basically you put all these attachments within the experiment so that everyone is at the same page in terms of information about this experiment. That's very powerful. Next is sharing, right? Cytobank platform is a tool, collaborative tool in a sense, because you can create your experiment, set it up with like these labels, do illustration, but then you can share it with your colleagues. And this takes just a simple click. So when you go to the sharing part of the interface, like shown here with the red arrow, then you can just select the username of your collaborator. And once you've done that, instantly we'll have access to all the work and documents that you have associated to the experiment, which is very fantastic. Next, you can also share at different privilege level, and this goes through the use of a project and definition of a project. So to set sharing privilege, you need to create a project, and then you have access to this window where you actually can define project members as managers, so they have all rights to modify and do whatever on the experiment level. Leaders that have a little less rights, they can share and clone experiment. And members can only visualize data, they cannot modify anything. So that was for the experimental manager part. Now I wanted to highlight a few things about the gating editor. To show you that it's just not about gating. So the interface within Cytobank is actually powerful for other things. So first, how does it look like? So that's the gating editor interface. We see that there are several windows or several interaction boxes. One shows, for example, the gating tool. So that's quite common of what you can use as a tool for gating your data. You have a way below to change the analysis variable, X, Y, and also Z, depending on the plot type you select, the red box. And then you can either define gates on the orange box on ungated data, or you can change the parent population and then set a gate on this parent population. So that's very nice. So you can gate this from scratch, but very importantly, you can also upload historical gating strategies from previous experiments you did in Cytobank using the exact same panel by this simple button import gates that we see on the header of this interface. But we also can importantly use gating ML, which is the standard that has been defined by ISAC to actually allow data curation across different platforms, different softwares, and also this is a standard that is being understandable by bioinformaticians using R. So it's really key, that allows you to basically validate results that unsupervised analysis may have done on a traditional gating strategy. And all this is possible through Cytobank. Now, the other nice thing about Cytobank is the data scaling. It's very important to do that, because if you don't set your scales around 0 correctly, you might have issues at dimensionality reduction techniques. because basically the algorithm will see, for example, if we see arcsinus H without any transformation, you see some cells that are very negative, and some cells that appear dim, and some cells that appear high. Actually, it's a transformation artifact, but the algorithm will separate that into different populations, which are artifactual. So you really need to set scales, and that's why when you create a new experiment in Cytobank solution, the first step that one is asking you is to adjust scale. That's very critical, and you need to do that. So, but what I wanted to highlight here is that the scaling tool within the Cytobank interface is very easy to use and also cope with different data types, so flow, mass, spectral, and genomics. So you can scale all these data types with the transformation included in the tool. And you can also work on data from different flow cytometers. So we tested several ones because we do benchmark of flow cytometers also as one of our activities, and we actually checked all data types produced by different instruments on the market, and they were always you could always analyze them on Cytobank and scale them properly there. Next, what I wanted to show you is a very, very cool possibility, that is to be able to derive gates from enriched parameters or cluster parameters. So for example, here we have single cell genomic data set. I'm looking at UMAP dimensions. And what I would like to do is basically here I project Hervé data, so that has been enriched into another software not available in Cytobank, but I can visualize that, and then I see my color code that fits to cluster annotation. But what I would like to do is to calculate frequencies of cells within each of these clusters. So for this, I can create an automatic cluster gate by pressing here, selecting my dimension where I have the cluster information, Hervé, for example, here, and then apply these changes, and automatically Hervé will segment the data based on the attribution of each single cell to this given cluster. And that's really powerful because now I can use that for subsequent analysis. In the same time, it creates population that are all regroup all cells belonging to each of the clusters. And here I have the identification in this cluster table. I can look at the frequencies using the population tree and see what's the weight, relative weight of each of these in my sample. And then I can also visualize it through something I like very much, this visualization through sunburst that shows you the different proportion, relative proportion of each cluster. But I can even push further. I can also split my file. So in it, I had two groups, basal and inflamed. So I can actually use that sample flag that is included in my FCS to actually create new files. So for example here, that's how I can clone an experiment. And then here on the bottom, I see I can select populations, and I will select the population that relates to my sample flags. And what Cytobank will do is basically split the files in two and allow me to investigate the same gating strategy or the same thing I've been doing before, and then visualize the result. And like this, very graphically, I can see that in my inflamed condition, I have this cluster in gray that is increased, and the cluster 6 that is decreased in my inflamed group. And then I can directly relate it to percentage. So graphically, it's very, very powerful and efficient. Then what I can also do, so I've kind of already shown that. In Cytobank, you have these machine learning tools that are embedded, actually already integrated in interface, which are FlowSum. And you also have Visni, you have additionally Spade and Citrus. So all these tools you can use in Cytobank, but you know, the pace of the algorithm development is really fast, and there are basically every month a new method that comes out. So how do you do to actually still use this nice tool that is Cytobank with these new methods that comes? Well, that's what we did at CIPHE. Basically, we at CIPHE we can take the FCS file that has been analyzed first in Cytobank. Cytobank put the new coordinates for clusters and also dimensionality reduction into new parameters. So it enriched the FCS file with this information. And then I can use these files to do another analysis, like here scaffold. And then once this is done, information from this new method is enriched in the FCS file, and I can log it back to Cytobank and do a final visualization and share that to my colleagues. So that's something really important we do at CIPHE. Just here about the concept of FCS file enrichment so that you understand well. So you start with your matrix of parameters where you actually have each line a cell, each column a parameter, and then the value measured from the instrument. What you can think of doing is dimensionality reduction using these variables here. And then what your software most of the time do is that you can actually compute this and then integrate the new information into the original data matrix. So you enrich it with either dimensionality reduction coordinates or clustering like flow sum here. And then once this is done, you can add more and more, do more clustering, and each time sequentially enrich your FCS file. The really cool thing is that each of these new parameters are available for Cytobank to be used for visualization, or like what I showed you before, splitting data or creating population from this new dimension. So it basically increased drastically the capacity of what you can do using your cytometry analysis. And then the end goal is always visualization on Cytobank because of the capacity to keep the cellular level. So you can project your information at the cellular level, and that helps you to actually QC your data in the clustering steps. So here it's another example of that we did in Cytobank. So we projected Hervé clustering results onto a t-SNE map. Actually, it's an opt-SNE map. So something you cannot do now on Cytobank, but you can use the visualization techniques that are embedded in it and that are very efficient. Now let's shift to something that is super cool, very important for me. It's the illustration mode, because there is, when you have 156 files and you need to analyze that in a comprehensive manner and share the information to your colleagues, you need something else than just conventional flow cytometry software. And this is what Cytobank really brings us. So that's an example here of an experiment, immunophenotyping of several inbred strains and mutant mice on these given genetic backgrounds. So you have 16 different backgrounds, and then starts what to do with that. So first of the thing you should think about doing is actually to annotate your data with sample tags. That's very important because that's the power actually of what you could do in terms of analysis later on. It's coming from that. So for example, you can do that manually, and I have put the link of the manual annotation here. But you can also do that automatically. So the easiest way I wanted to show you is through this. So you can first go into export sample tags. Then you have the predefined tables that Cytobanks use to actually encode the different conditions that are available in the platform at start. And then you can basically create your own tag and re-upload that sample tag into the platform and then have access to it. So it's a very convenient way when you have a lot of files, so here is 16, but when you have a plate, for example, to be able to annotate your full data set in one click. Then what you basically have is you use this sample tag next to actually drive your illustration design. So in Cytobank illustration mode, you have different boxes, and here it's the sample tag box. And you see that I have set up condition tags that actually I use that to put the information about the mutation that I'm looking at. I have tags about experiments because the data have been acquired across 2 experiments. And then I have group tags for the genetic background. So like this, I can basically filter using these tags. I can filter my data set and only show a part of it. So what I'm showing here, although I had mutants on BALB/C background and NMRI background, I have filtered all the other mice, and I'm only keeping the ones that are on the B6 background, and then I am showing all the mutants that were acquired on this B6 background, and then you see nicely the results. So it's very, very convenient to actually adjust what you want to show on an illustration using these tags. And I've seen a lot of people not using this capacity, and I think it's a big mistake. That's a very powerful functionality of the platform. Another example I wanted to bring for this webinar is actually the capacity to change the statistic that is being shown on an illustration very quickly. Like you have one question, you are reviewing data with your collaborator over a teleconference, and then someone asks suddenly, so you showing here a percentage of interferon gamma producing cells in the different in these different populations, but how many cells you actually have? How does it relate into cell count? So usually, in traditional approach, you will need a secondary analysis. You will exchange with your colleagues back and forth, and then solve this question. Here in Cytobank, directly over the TC, you can basically change from percentage to event counts, and then directly the results are actually drawn onto your illustration. So it saves you time on these finicky details and allows you to move forward into your hypothesis building that actually this teleconference needs to be. It needs to be a time where we share information and where we actually can make proposition to move the project forwards. And that's what I really liked about the use of Cytobank in this way over collaborative TC we may have at CIPHE. The last thing I wanted to show is, because I've seen lots of colleagues that don't do that too, so if I can bring that, there are ways to save your illustration. So you can really design several different illustrations that you plan to show to your colleagues. You can save them and then show them later during a TC, or allow your partners to actually go and consume this visualization that you have prepared for them. You can also use templates. So if you are really fan of one given visualization way, you can actually click on a button, and then automatically the template will be applied to your data set. So it saves you time in doing, if you are doing over and over again the same kind of analysis template illustration, then you can do that very quickly through this capacity. Then, for example, you can, like this, create high-quality figures. For example, here I project my marker expression onto a Visney map to check where are my CD4 expressing cells, my CD5 expressing cell, my CDX expressing cell. So it's a mass cytometry data set. Then I can do also a panorama very quickly by actually increasing the list of marker I want to look at, and so that also I don't get lost. I can order that by alphabetical order or whatever order I want by a simple click and drag action on this window. And again, that's something seems very, very little, but it is really make this picture, this global picture, a lot more readable from start. And the final thing, you can do basically a crosstalk between different visualization. For example, here I have one question. I have made clusters. I can visualize the expression of markers on heat map in Cytobank, and then I can also project this information onto a T-SNE map so that I can relate where are my cells that express this and this marker. This could be the cell type, and then I relate that to a density plot that is shown on the upper part of this figure, and that's basically linking this free visualization that allows you to have an understanding of what's going on in your sample and to drive this information efficiently to your partners. What I also wanted to show you is that the tags, they are not limited. So basically, if you want to create a new one, you have the possibility to do that through this add button. And then you basically create your new category, and then you can order your FCS file based on this new nomenclature that you are creating. For example, here, I created a metal barcoding tag so that I can encode each file corresponding to each debarcoded or barcode that I was using mass cytometry to encode my samples. And then I can visualize whether the debarcoding was okay, or if it's okay, I should see always 3 yellow box and then 3 black box. So like this, I can have a feel about the quality of the debarcoding, and that's particularly important if the work that is being done is actually done over several people. So you might have an operator that do the debarcoding and someone else that takes into responsibility the advanced analysis. So you need to have some ways to check that everything went fine at start in the data set you're analyzing, and that's one way we do at CIPHE to check that debarcoding was okay. So you see, you can drive this heat map very, very simply by changing the plot type to heat map. And then you can also display different kind of metrics. So here I did a ratio, took the minimum of the column, and then I show the mean of the channels variation like this. So you can change all this. So it's very easy to actually change the way the heat map looks depending on your question. So next, what I wanted to show you is another usage we have at CIPHE. I have bioinformatician people that do develop new methodologies. So for example, here they are working on using deep learning to actually annotate samples. So they are building different deep learning models to annotate the same data set, and we compare that to a data set that was annotated with the scaffold algorithm. And what you can see here is that each time I'm projecting either the cluster identity from scaffold or from the one that were given by the perceptron that they use in these deep learning models. So that was showing total cells. I can now select, because if I have done it before, I can identify my neutrophils by conventional gating, and then I can show only the cells that are within this population in my plot and see whether the annotation is homogeneous or whether I have one method that does not do a good job. So here we see it's each time blue, so it's each time the same type of cluster ID that is being shown. So I have a equivalent resolution about my method, about this annotation on neutrophils. Now, if I look on T cells, I see I have several colors. Well, that's fine, because CD4 T cells have different functional subset, and they could be set into different clusters. But to investigate that, I need to change the dimension I'm looking at. The t-SNE are not good enough, so I can very intuitively change it on this axis now in the new interface of Cytobank, and dynamically I can adjust what I see, and I can see whether the clustering is performing okay, or how actually this work has been done by the algorithm. And you can already see through this way that scaffold, for example, is not segregating the data like perceptron models, that they do more defined based on the training data set. They do more defined thresholds on the data. But that's basically one way. So you can have your bioinformatician that load new data sets, that newly analyzed data sets onto Cytobank, and then like this way allow you to actually visualize and QC what has been done and bring your biologist perspective onto the new methods so that you can improve the results of these new methodologies. Right. So that's a very nice way to work between two expertise, right? Bioinformatician and biologist. I have to say that there is an API that we don't use at CIPHE, but that allows also bioinformatician to run analysis on Cytobank directly from R. Then the final things I wanted to show is more on the how to actually use Cytobank to export data. So it's not only a visualization interface. You can actually export a lot of different descriptive statistics that are embedded within the tool. And I wanted to show how to do that, and also how we can use this advantage of the having a figure or an illustration that is driven by sample tags to actually very quickly generate a figure for a publication quality and format through Cytobank. So for the results, the export of the result table with the sample tags, basically you go at this position in the interface, so export statistics. Then you come on this window where you basically can select if you want to add a statistic or sample tags or keywords into the table of results, what type of statistic you want to export, and there are a lot of them. If you want to export that on the segment or some population, and what is the parent population. So you can define all that, and then each time it will add columns onto your table of results. And then you can export that. It comes into basically a CSV, a spreadsheet that you can open to Excel. And what we basically do at CIPHE is that we just build very quickly an Excel table and then use pivot table to make aggregation and calculate average percentage, for example, of T cell across the different conditions. And that's possible because I have embedded into one export file the sample tags as well as the results of the percentage of population that I selected at the data statistic export. So it saves you a lot of time because you have all information in the sorry in the spreadsheet that you can use for further analysis. Then obviously these tables you can use for third-party software to run statistical analysis. So this is up to you, the tool. Then about the export illustration elements for publication quality. So for example, here you can, through the layout of the plot, you can change the size of the plot that you are showing. So here I have taken the highest number of pixels per plot, and then what I did is that I can, on the export button here, I can change the type of export. If you go in export option, you have several choices of what you can have. So I've been minimalistic. I only wanted to have my plots, basically. I saved it in vector graphics so that I can open it straight into Illustrator and then be able to edit my text and then have my figure arrayed already for the publication. So that saves you a lot of time. You don't need now to align and array each individual plots. Everything is really positioned at the right place from start. So that's all for the capabilities I wanted to show you. So these are, again, different than regular webinars you might have on Cytobank, where people focus on the advanced analysis tool and machine learning tool that are embedded within the platform. But I think it's not only this. So you should. I hope I could show you that through my presentation, that there are a lot more than just machine learning tools into this solution. So as conclusion, if we want to go back to basically how we use Cytobank at CIPHE, so there are different branches. There is the internal part at CIPHE, so where we can basically have biologists that run analysis tool, either at CIPHE or on Cytobank platform, and then launch or upload the enriched FCS file onto Cytobank and do metadata-driven illustration, store data, annotate, share that over a consortium member like Sphelfedia. And then for the feedback we get from this published data / illustration, we can go another round and maybe do another type of analysis to actually enrich the experience of the data set that is being stored on Cytobank. The other part is the development of new methods and the validation by biologics, by experts of the method. So it's not only using metrics like F1 score, but really also using the expertise of biologists to curate these new methods and see how they behave, so that bioinformaticians and biologists have together a better understanding of the method and try to improve its application. And Cytobank is a great exchange platform for that within your team. And finally, Well, here I've just listed the few ones that could actually beneficiate from our intensive use of Cytobank over this COVID sanitary crisis. So we were locked down, but actually data analysis were progressing thanks to Cytobank. So we could do data review, fine, we could do reports, we could generate illustration, figures for papers, and so on. So that did not stop us working, basically, thanks to this cloud-based solution. So just a few lines to sum up. I hope I showed you and convinced you that Cytobank is a platform that fulfills by essence these FAIR principles that are more and more asked in the field of data science. So your data should be findable, accessible, interoperable, and be able to reuse the digital data sets. If you actually combine sharing data experiment on Cytobank using label tags that are descriptive enough so that people can find themselves through what you actually did, you really are into this fulfilling this principle. Now, since there have been a lot of work by Beckman Coulter and Cytobank to improve the interface, so I find the new interface really nice because it allows fast adoption by new people. The experiment management tool is, as you have seen, really improved the efficiency to access the data and find really important experiments. You've seen the use of illustration has been rethought, so now it's very easy to actually design illustration on multi-group, multi-samples, and interrogate part of the data set like this using like dynamic filters. You can share data very quickly as a click, and then I've mentioned that, but you can really do this interactivity, this change of illustration, and so on. You can really do that over TC. So because the server response pace is quite good, so it does not take long so that illustrations are updated. And like this, you can, as I said, solve technical questions and focus your TC time with your collaborator on advanced question and building hypothesis, which is what we should really use this time for. And finally, because we no need to perform offline secondary analysis because we really solved all these little issues through the TC. So I'm now done. I wanted to thank Beckman Coulter for this opportunity to present you this capacity of Cytobank and the way we use this software solution at CIPHE. And also I wanted to thank the people involved in this work in one way or another of what we do every day at CIPHE. Thank you very much for your attention, and I'm happy to take questions.
Thank you, Dr. LUCHE, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is, how do you deal with license management and training at the level of a consortium?
Okay, so for the license management, we basically use a Cytobank that is called Premium Cytobank. So all the license management is actually not done by us, but it's done by the Beckman sales rep, which is saving us a lot of time, which is, for me, I think it's really good. And then you have to plan in advance the license cost in your project. So like for Selfedia, when we applied for the project, we applied for money for the license of Cytobank for 1 year. This got extended for a second year due to the COVID situation. But basically, I had really no issues getting this extra money because the managers of the funds, they understood very well the advantage of having a Cytobank platform to actually share data through the consortium. We do guide new Cytobank platform adopters through face-to-face trainings, could be through a teleconference or desktop sharing, but we also advise them to ask in parallel for support because Beckman has actually built a team, an application specialist team that can help you in going through some particular aspect of Cytobank. It's very rich interface, so you cannot show everything in one webinar. And finally, or one face-to-face action. And finally, there are plenty of support material, videos, webinars. There is also a blog that demonstrates the different capabilities of the Cytobank platform. So I really encourage people to go and look at this to train also themselves on the new interface.
Thank you, Dr. LUCHE. Our next question is, how do you manage cross-data validation of advanced analysis with historical data?
Yeah, so that's a very important point, because when you do these collaborative projects, then you basically have to bring your expertise, and so you are bringing maybe like for us it's high content parameter analysis and in mass cytometry or spectral and flow, and using advanced algorithm methodologies. But our partners, they are experts in the biology. They are experts. They already did preliminary experiments. And they need to share us the data, and that are the fundamentals of the collaborative work that we will undertake. So really using Cytobank to ask people to actually share their historical data set on it is really important. And then next also, because we have now access through Cytobank to this historical data set, we can also, as I explained, we can take the gating strategy that people are using and project onto this gating strategy annotation or cluster analysis that helps them this time to actually better understand these advanced methodologies. So you have a link with historical data, so it makes it very much easier for your mind to understand what's going on and what is being done. And also have confidence in the analysis that is driven in a supervised manner. So gating ML is the final one. Also using this standard gating script, you can cross-validate data.
Thank you, Dr. LUCHE. Our next question here: why use a cloud solution for data analysis instead of a desktop analysis tool? Well,
I've just talked about this a little bit. When you are in a collaborative project and you have several people that are implicated, it's actually a limitation to work on your workspace because you spend a lot of time to build PowerPoints, and this is all rigid because if you forgot to address one question or that your collaborator may have, then you have to go back to your workstation and drive a given illustration to solve that. As I've shown you on Cytobank, it could be done on the fly. Then it's also very, very nice because I have my way to analyze the data, and maybe that won't fit the way of my collaborators, but because I share data set and illustration, they can, with their license, actually reanalyze the same data set their own way, and then we can discuss about what overlaps and what not, so that we are all at the same page. So for me, this is a lot actually more rigorous, because like this you get the expertise of several people looking at the data set. And for example, in the Self-EDIA consortium, we actually did build a very big rat panel, but we don't have any knowledge about the rat markerology, I would say, what type of cell express, what type of marker. And like this, doing the analysis collaboratively on a cloud solution allowed our collaborators to actually look at the data themselves and provide feedback, which I think is really the strength of the platform. Final thing is that if you run machine learning methods on a lot of events and a lot of files, that takes a long time for your computer to compute. And if you do that on Cytobank, basically your computer can be used to do something else, because you are using the server CPU to do this analysis and not your machine.
Thank you, Dr. LUCHE. Our next question. What is the most interesting feature of the Cytobank platform in your views compared to the others?
So definitely using metadata tag to drive illustration, the way I can plug my metadata describing my sample directly with the FCS file using an Excel spreadsheet, that's super convenient because you can have lots of meta tags that you want to use, and in different methods you have to add keywords. I mean, it's quite laborious. Here it's very easy to do. Then the capacity to export plenty of descriptive statistical measures that can be used in third-party software is also really nice. So you don't have to compromise because the possibility you have is quite big, quite large. And I also like the features that I have tried to highlight here about data management level, that I may have people that only come and consume visualization, some that will have rights to actually clone experiment and do their analysis on their own and edit graphics very quickly. So this is very, very nice to have, so that the work basically can be shared through this capacity across the team.
Thank you, Dr. LUCHE. And our last question, for which type of cytometry do you use the Cytobank platform the most?
We've been early adopters of Cytobank solution initially for mass cytometry dataset because we had no bioinformatician, and that was at this time pretty much the only platform that allowed us to do efficient analysis of the new data type. I have to say now we are not restricted because we actually do a lot of flow experiments, loading them on Cytobank, also spectral, and we also did some multi-omics data set where we look at protein and gene at the same time. It has to be, there are some restrictions. It's limited to 800 variables, but this is nice because it allows again biologists to be at the field of the action and drive their own analysis. So I would say it's not restricted now to mass, and I really think that it's a good advantage for flow and spectral analysis, especially if you have lots of files. What we have tested basically is that the solution is compliant with lots of different conventional flow cytometers. So each time FCS file format for a provider to another one may be a bit different. So Cytobank solution is very reliable, and it's basically a sort of Swiss knife, so you can use that to look at whatever data set generated on this instrument. And to conclude on that, I would say that nowadays we mostly use Cytobank solution to project our data analysis capabilities to our partners due to this very easy sharing capacity. Like this, we allow our partners to have access to opsonize raw data set through the illustration interface of the Cytobank solution. And last but not least, within the CIPHE team, I often ask my collaborators to share their just acquired data, when there is something really burning that we never looked at, so that I can have a look at it too. In a remote manner, I can even, if I'm traveling, I can look at even on my phone and then give suggestion or advice from what I'm seeing there, and that's keep you in touch with the data that is being produced wherever you are. So this is very, very nice capability too.
Thank you, Dr. LUCHE. Do you have any final comments for our audience?
Well, I hope for this webinar I managed to bring that Cytobank is actually more than just machine learning tool. It's really allows you to efficiently dive through your data sets and also communicate with your colleagues as either bioinformatician or biologists of different areas. So that you come across together faster, and you can build hypotheses faster, and then be more productive basically in your analysis scheme. So I hope I managed to do that.
Well, thank you again, Dr. LUCHE, for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Bye.